Hi, everyone. My name is Wei Sun from University of Buffalo. It's very pleasure to be here to share my research project with everyone. I want to thank you, Hyperacusis Foundation, and also all the support to this research. Today, I'm going to share a research I recently studied. Is using Fox gene syndrome, a novel model for hyperacusis study. We know, I hear lots of story. Lots of people's hyperacusis was induced by noise exposure. However, there is other group of people develop hyperacusis based on audiological developmental disorders. For example, Williams syndrome, autism, and I'm talking about today is called Fox syndrome. The reason I started to study this syndrome is one of my colleagues. Her daughter have these symptoms. And at the very beginning, she didn't know what it is because she studied genetics. So she starts to diagnose her daughter's symptoms by genome screening. And she find her daughter have one very rare mutation in one gene called the Fox gene. And turned out not only her daughter had these symptoms, he find thousands of parents also have their children have the similar gene mutation. The major symptoms is a seizure and motor disorders. Lots of children you can see here have to sit on wheelchairs. And also there is speech disorders. So they have overlap with lots of autism symptoms. So she developed this animal model. She want to screen the seizure drugs to cure their disease. Then when she had this model, she sent to me to test if they have audiogenic seizure. Audiogenic seizure is very rare, so it can be triggered by the sound. So some animal model we know in rodent can have this seizure. Another name of this disease is called microcephala, small brain. So the children have small size of brain because they have developmental problems to cause these symptoms. And I studied this audiogenic seizure in rodent, and I didn't find I have very rare of these mice do have this seizure, but I don't believe it's a cause because I know rodent develop audiogenic seizure is merely by the inferior colliculus. And this developmental cause in the brain is the auditory cortex. And human have audiogenic seizure is in the auditory cortex. And then I asked Dr. Lee, and he sent the questionnaires to their foundation to see if their children have hyperacusis. And I do find some patient, about 13 patients sent me back of this questionnaire. 13 of these subjects is the only parents report their children have a hyperacusis. Two of them, lots of children, parents report their children have to wear earmuffs when they go out, otherwise they're going to cry. Two of the parents report their children can have a seizure if they hear a loud sound. So I thought this could be a model to study autogenic seizure. This model also have lack of the corpus callosum, the white matter between two brains. You can see this is the normal one. This is the mutant mouse. So I study one of the mouse have the minor symptoms for the behavior because some of mouse when they were born homozygous, they have two pair of mutation, they cannot survive. So I use only one of the mouse to try to see. So two aims I trying to study. One is to see how to access the sound sensitivity and tolerant in this mouse model. Because hyperacusis is not only a sensitivity issue, it's a tolerant issue. Second thing I wanted to see which part of the cortex, because I think probably the cortex is developmental issue, contribute to this tolerant reduction. So people report Fox gene also expressed in cochlea. So I first want to find out they have a hearing loss. I do not find hearing loss in this model, which is consistent with Dr. Lee mentioned because very few of the children report they have hearing loss. They didn't notice that. I think this model is, for some reason, for me, I think is a plus because if they have hearing loss, then I don't know if the hearing loss causes the behavior changes or is the hyperacusis caused hearing loss. So this model, they don't have hearing loss. So everything we compare the wild type and the homozygous should be hypoacusis or central part of the brain contribute to the sound reception, perception changes. So first thing I did the startle reflex because it's a simple, not require a training. Opposite than the noise-induced model, they don't show enhanced startle reflex if you just run 20 trials or 30 trials. However, if you run long-term trials, like 100 trials, we do find in the beginning of the trials, the startle did not enhance, but in the end of a trial, they do enhance. This suggesting it's a habituation issue because a lot of times when you hear a sound, gradually you will ignore the sound. You can habituate to environmental sound, but this mouse seems to could not habituate. Second thing I find out is they have a very poor gap PPI. Gap PPI is a gating effect. So for example, we can ignore some of this response, but since this mouse cannot ignore, so this seems is a top-down temporal process. You can also interpret it as temporal processing. So this is a knockout mouse, is a control mouse. This is also opposite in noise-induced hypoacusis model because noise-induced hypoacusis model, if they don't induce severe hearing loss, you have a better temporal processing. Then we try to evaluate the sound tolerant. So we do the open field study. So we leave the animal in an open space. We watch how they move. We compare the quiet and the noisy environment. Noise, we only use 90 decibels, is not very long, very high. First one we notice the mutant mice, they move more because the people already know they have the motor disorders. The mutant mice, you cannot raise them into same cage because they're going to fight. They have the what I call it, they have the hyperexcitability, also have this aggressive behavior in this mouse model. This is also another symptoms for the autism. And when we expose to sound, and those mice, the mutant mice, immediately stop. So they freeze. So you can see this. They trying to stay in one place means they freeze in one place. Then sometimes they run, they jump, seems they are scared to the sound, even 90 decibel sound. So I think for the preliminary data we find out is they do show some tolerant issue, even it's not, may not be hypersensitive to the sound, since they cannot tolerant to long-term of sound stimuli. Then we trying to do cortex recording. So we do the multi-unit recording, put it in the brain to see the cortex response. We expected if they perceive sound loud, they should have an enhanced cortex response. This is what also central gain of noise model showed, but we see opposite. You can see the green line, blue line actually is a wild type mice. Red line is the mutant mice. We say, okay, how we interpret this? They have lower tolerance to sound, but they have lower response to the cortex. But we also noticed, so the response after the onset of sound, they didn't go back to the baseline. Originally, we think that's very minor, but then we try to do another group of mice. We do the wild type mice. We did no exposure. We try to mimic if the central gain was also happened in this group of mice. We do find enhanced central gain. This is the same wild type mice. We just expose them with the moderate sound for 2 weeks, 85 days, 83 decibels for 8 hours per day. They do show enhanced cortex response is fit to the central gain. But you also find out, oh, I didn't show that picture. They also going back to the baseline. So even the minor change, we do see this exactly opposite of the cortex response. So we think this post-sound stimulus response probably reflected to the mice cannot habituate to long-term of sound stimuli. This is another analysis called the current source density, trying to see which layer of the cortex actually contribute to this hyperactivity or hypoactivity. So this is a control mice. This is a mutant mice. You do see they have light colors, which indicating there is less response in the different layers of cortex response. Opposite to noise-induced model. Noise-induced model consistent with enhanced AC response show much enhanced color, which means there's a hyperactivity in the cortex. Because central gain suggesting because the brain have more activity is because of reduced inhibition in the brain. So we're trying to look at inhibitory neurotransmitter and the excitatory neurotransmitters. So left panel is control, and the right panel is mutant mice. We do see both reduction of the inhibitory and excitatory neurotransmitters. So we think this model, because it's developmental disorder in the cortex, so they do show reduction of GABA and glutamate receptors. So this is just preliminary data. So this is generated our hypothesis. We think the noise-induced mouse model or hyperacusis model may be triggered the enhanced central gain, which we already see in noise exposure model. And the other side of the hyperacusis caused by the brain development could be reflect either autism or Williams syndrome, or maybe this Fox gene mutation. Maybe it's causing the lack of adaptation to the sound. So even the sound, short-term sound is okay, but long-term sound, the brain cannot tolerate. So that can cause the reduced sound tolerant. So that's our hypothesis. So we plan to do the in vivo study to do the wake recording in these mice, trying to prove our model is correct or not. This may not reflect to everybody's, it's about especially our patients' symptoms, but this mouse model gave us another view, what can cause people have reduced tolerance to sound and affect their sound perception. Okay, thank you.
Right. So a couple of questions on the immunohistology that you showed. So you talked before that the homozygous mutations are lethal, but it looked like the data was from homozygous tissue.
This is a single nuclear mutation. They are
Okay.
not lethal. So
Okay.
they can, homozygous and heterozygous can both survive.
Gotcha.
Mhm.
The drop in VGLUT1 is drastic. Do they have like a concomitant upregulation of 2, 3? Like there has to be like some form of glutamatergic neuron and vesicular loading there, right?
Yeah, that's a good question. We haven't looked at other type of glutamate receptors yet. We did look at different layer specific staining. We did other staining I didn't show here. They lose the density of the neurons in both super layer and the deep layers. They do, but we haven't looked at other side of the glutamate receptors.
Yeah. I mean, to release it, right? They have to have some type of transporter to be able to put it in and package it. Okay. Thank you.
Thank you.
Just one question, comment on that. It says fantastic job.
Thank you.
So we've just heard from our current recipient of an emerging research grant. And now we're going to hear from the previous occupant of that role, Megan Wood. Here's the talking stick.
I normally have a very loud talking voice, so I'm going to try to modulate a little bit. I'm going to hopefully, where am I pointing? No,
Not working?
I'm clicking. Where am I pointing it to? Your computer?
Yeah, right on.
Oh, okay, I went back. There we go. Okay, great. So in the second year of my emerging research grant, I really focused on an idea we'd had for a while, which was trying to develop some sort of behavioral assay to show that mice could experience pain to sound. And so today I'm going to show you a brief portion of the poster that we had yesterday that my undergraduate presented, where we've made some progress in this space. The first main question was, can a mouse, they're exposed to 120 decibels of sound, which is about the human threshold for pain to sound, would they create a facial grimace that would show us that they're experiencing pain? Okay, listen. Yeah, great. So, yeah, perfect. So, what we did is we took 6-week-old C57 black 6 mice. So they shouldn't be at the level where they have overt hearing loss yet. We videotaped them using a GoPro in these custom polycarbonate cages, which were clear, these 2 at a time. And we had these set up in such a way where there were air holes on either end. So the mice were mostly in profile, and that was really important for this. Facial grimace has been used to assess pain in humans and many different rodents, but often the way it has been done in the past is face on. And the way that then that was analyzed was to take the clearest video frame from 2 minutes of video and then to train someone that was blinded on different features of the face, 0, 1, 2. Are they experiencing something associated with pain or not? I didn't like that approach. I felt like that could introduce a lot of bias in a lot of different ways. So there was a paper that came out recently where they did a similar, they kind of made up these new ways to mark the face and look at facial grimace parameters, but from the profile of the mouse. And but they put all these markers onto video frames manually. So Anna was willing to try to use deep lab cut. And so we used the same markers that have been published to put markers all over the face in different parts, the eye, the ear, the nose, the mouth, and have deep lab cut mark our entire video of 40,000 frames. And then we could pull out the data in a more unbiased manner. So the way we actually did the sound presentation, which has some issues, but we're going to keep working on this, is we had 10 minutes of no sound. And then starting at the 10-minute marker, the mice were introduced to 70 decibel broadband sound. And that was increased by 10 decibels every 2 minutes until we hit 120. And then we turned the sound off. And what I hope you can appreciate along the bottom is that on the left is a mouse in profile experiencing no sound, and he's fairly happy. In the middle is a mouse experiencing 80 decibels of sound, eyes open, ears are alert, looking fairly normal. And then the mouse over here is experiencing 120. And you can see a very noticeable change in the facial features of the mouse. The ear is back, the eye is squinted, the nose is rounded. And so we then went on to quantify those changes. Cool. So like I said, I'm not going to present all of our data, but I'm just going to focus on the eye because in facial grimace literature, that has been one of the markers that has been most associated with pain. So the way we do this is we have 4 points in the eye, and then those axes across the eye are used to generate a ratio. The smaller the ratio, the more the eye is squinted. On the top left here is a trace of one video from one mouse of the entire 25 minutes. And I hope you can see, I'm sorry for the people on the side. So there's silence. This is 70 decibels, 80, 90, 100, 110. And then you can see how the eye ratio drops at 120 and then it recovers some after in no sound. So that's one mouse though. Across the bottom is 12 mice that have been averaged in the black lines. And you can see again, our no sound at 80 decibels, the eye is opening and closing as the mouse is moving around and just behaving like normal. When you get to 120, you see a drop in the eye ratio, and also there's less variability because mostly the mouse is freezing with its eye closed. And then very interestingly, when you turn off the sound, there's a hangover of almost 30 seconds where the mouse doesn't recover until they start acting normally again. So then we had to kind of look at all of our data and try to figure out and all of the other measurements we made, because we made many different measurements of the ear, as I said, the snout, the mouth. We had to try to come up with an idea of what our definition of pain would be to be able to continue doing this research. So here on the left is looking at for each sound level. It's every single video frame that was analyzed, and that's 12 mice laid on top of each other. So you can see the overall trend in the data is actually that at 70 and 80 decibels, the eye opens more than normal, probably because they're just hearing sound. And then over the course of the sound presentation, until you get to 120, the eye is closing and closing and closing and closing, and then it pops back up some in the post-sound time. The middle is just the average of all of our mice, all of the frames per mouse as the single dot in no sound, 80 decibels and 120. And then on the right, the first way we've kind of tried to think about this is what is the change from baseline being no sound for each of these sound presentation levels. And here you can kind of see that a negative percentage change from baseline is going to be not painful, and a positive change from baseline is going to be painful. And at 120, you get almost 10% change from baseline. When we added all of our different measurements together, one thing that was interesting that we found was that in aggregate, again, adding all of these different percentages together, which is kind of an odd way to do it, 120 had a 30% change from baseline, mostly driven by the ear and the eye measurements. And 110 actually had the same effect. So that kind of tells us that we probably don't even have to ever expose a mouse to 120 decibels. 110 would probably be enough. And then there was kind of a graded response where 90% or 90 decibels gave us about a 10% change, and 100 gave us about 20% change. And so it was kind of an interesting way to look at the data and think about going forward. If we have mice that are experiencing pain, could it be even just a very slight change in pain, or could we be able to capture that basically with this type of assay? So you can move to the last one. So where I'm kind of going with this, I personally am going to continue to work on noise-induced hyperacusis. I think that that's really interesting based on some of the patient data. That is going to entail a lot of work trying to figure out the correct noise exposure that is needed to induce hyperacusis. In some work that has been previously done by a PhD student, after noise exposure, only a subset of mice develop hyperacusis. So we may be looking at something kind of like that, where we can compare in noise-exposed cohorts, hyperacusis-like to not hyperacusis-like. One other question is, obviously, the mice need to be able to hear sound. So we can't noise expose them to the extent where they have a huge permanent threshold shift. So it would be better to start with a synaptopathic noise exposure, which also may replicate more of like an before overt hearing loss in humans type of a model. So I'm really planning to do that. But the other reason that I wanted to kind of develop this assay was as a tool for the field, because obviously we've heard about several different genetic mouse models or mouse models that may be associated with hyperacusis. Tianying, who's here, was doing the GJB2 het. And then we just heard about the FOXG1 mutation. Ben Auerbach has done Fragile X syndrome. But this would really be a tool maybe for different people who are looking at genetic causes to see if there's ever a pain aspect. And then the last thing I just want to bring up is that, a lot of this and what I have depended on being a part of hyperacusis research and coming to some of these meetings is the patient experience has been really important. And I think that understanding more about the patient experience and also having a better outreach to audiologists and better data that we can pull from people who are actually experiencing this will help us model this better in animal systems, which hopefully would lead to faster cures, because we could then be able to really intervene in animal models. Thank you so much.
Any questions from the audience?
Have you looked into ultrasonic vocalization?
Say that again.
Have you looked into ultrasonic vocalization?
Oh, great question. No, haven't really gotten there at all or thought about it. I didn't mention, though, thank you for bringing that up, what the noise exposure band is. We're doing 2 to 20 kilohertz. So even if we do noise exposure, theoretically, it would be a low enough frequency that they could still hear it. But yes, what the animals actually produce and how that affects them would be fascinating. Even recording them after they've had noise exposure, or if they're in a hyperacusis-like state, would be really interesting. Thank you.
Hi, Megan.
Hi.
Fantastic type of efforts. Thank you, everybody. It's great we have these discussions, because that's the brainstorming how it comes to. And maybe unwillingly, I had some mouse models that could fit your purposes.
Right.
That was 7 years ago. We worked with a glassed mutant mouse model. So it has defective glutamate transporter in the cochlea.
Oh, cool.
Also in the brain. And it has synaptopathy. So half of the synapses in the cochlea are gone.
Mhm.
They're at all frequencies.
Right.
And what we've shown is that they're vulnerable for tinnitus induction
Mhm.
much more. So at baseline, they have normal hearing.
Mhm.
It's just they get more sensitive, and we used also gap detection to show that.
Mhm.
And one of the other phenotypes we had, but without really understanding because we were thinking about central gain,
Mhm.
we used manganese enhanced MRI. I don't know if you know
Okay.
about that.
Mm-mm.
But we used it in the area of pain
Oh.
research a lot. AstraZeneca has been done a lot of that stuff. Some mouse models work better than others. The idea is that you inject manganese chloride,
Mhm.
and where in the brain you get much more nerve activity,
Mhm.
it gets sucked up in the neurons, and you can use MRI to see the contrast.
Mhm.
And then those neurons that are more active shine like that.
All right. Mhm.
And glass knockout mice that are exposed to sound, which is a 90 decibel over 24 hours,
Mhm.
have their inferior colliculus shining like this,
Oh, wow.
not the wild type animals,
Mhm.
even if they're exposed to that amount of sound.
Mhm.
And these mice have also exacerbated startle response.
Mhm.
They're super reactive.
Mhm.
So maybe that's a model where you can combine synaptopathy and potentially also hyperacusis in it.
Well, and that's one other thing I wanted to say is that there's been a lot of work done with acoustic startle
Yeah.
and the amplification of acoustic startle, and I think that that's really important. And this would just be an additional assay to add because it would be
Yes.
fascinating if you had a mouse that had enhanced acoustic startle and no pain, or vice versa.
Yeah.
Mhm.
Yeah.
Excellent.
I have some embryos in the vials to send you.
Great. Sure.
Yes. Thank you so much for your lovely talk.
Mhm.
I was wondering, based on some of the stories we heard from patients today, it sounds like there's definitely kind of a progressive building upon itself.
Mhm.
And with this noise exposure model, are you planning to do kind of repetitive tests
Mhm.
with these mice?
So I think that there's kind of two components to that because that's something I've always been interested in. First is, do the mice develop hyperacusis or would they, and when? The previous work that had been kind of done in the facial grimace this space with sound, they measured the hyperacusis and behavior much later than we're doing because we're measuring it. We've done a little bit of noise exposure stuff and we've only measured out to like 7 days. So I think that even as we've heard tonight, that may not be enough because there could be a delay in onset of this behavior. And I think that chronic noise exposure is also something that's really interesting. One of the other places that I kind of want to go with this is to also look at different inflammatory models that might have some neurological effects like an MS model or something like that and see if that could also exacerbate symptoms. Mhm.
We're having technical difficulties over here. We just lost our Zoom chat person. But there's a question from Ivor for you, Megan. And
Can answer it here.
let's see if I leave here. There you go. He asks, does this framework represent reactive pain?
Does this framework represent reactive pain? This is more trying to understand whether the mice are in a state of pain is generally what we think of with facial grimace. It's not, so in other words, it's not pain to like the onset of a sound, it's ongoing pain while in sound. Is generally what we're thinking of more of this. Is that what he's asking maybe?
Well, I have no idea what you're talking about.
Right.
But I'm reading these words, and so he said, if so, and I'm wondering if I can just drop the question at this point.
Just go for it.
Because you said, but if so, do you see mice who become increasingly sensitive to lower sound levels?
Right. Well, we haven't done that yet, but that's kind of the next step that we want to do, because it would be interesting if we could ever find a noise exposure level, for instance, that would then, or noise exposure combined with a genetic model, let's say, where they would basically have lowered, I guess, increased sound sensitivity over time versus one that would just develop sound sensitivity and then it would just stay the same. But yeah.
And Ivor never asked just one question. Would that be required as necessary, though not necessarily sufficient, to indicate synaptopathy?
Oh, right. So I think that for synaptopathy, with the methods that we use, we would always couple something like this with histology at some point to prove that there's synaptopathy. I mean, it would be very interesting if they went together, but we don't know that that's the case. It would probably be, what we would probably do is measure this assay, ABRs looking at wave one amplitude, and then ultimately histology. And then it would be fascinating if that all went together, but not sure.
Thank you, Megan. Thank you, Ivor. Up next is Dr. Ben Auerbach. Just see if
better luck.
Yep. Is it turned off? Bad. Okay. We're gonna just have to.
Laser pointer works, but I'll just have to tell you next slide. Okay. How's it going, everyone? Thank you to Steve and Mike and Ivor for inviting me here today. I will tell you a little bit about my background in this. So I'm really a neuroscientist by training. I started off not working on hearing loss or hyperacusis or even the auditory system. My fundamental interest is in brain plasticity, how the brain adapts to changes in the environment, and that's how I ended up starting working on hearing loss when I did a postdoc with Dick Selby at the University of Buffalo. I'm sure many of you here know Dick. He was a mainstay at ARO and at these meetings. Recently retired, although he still emails me about once a month asking about papers that I need to submit still. So I don't know if he's ever going to really truly retire. So in any case, when I first joined his lab, I was interested in this idea of central gain enhancement and how hearing loss can trigger these kind of hyperexcitability changes in the brain, and how that might contribute to auditory perceptual disorders like tinnitus and hyperacusis. And so when I started, I had a really narrow view of what hyperacusis was. I thought, this is a loudness disorder that's triggered by cochlear damage, by hearing loss. And I think I've now grown to appreciate that it's much more heterogeneous and complex than that. Next slide, please. Yeah, so okay. All right, well, I guess I'll just keep on talking then. So I think now there's awareness that hyperacusis can encompass much more than just changes in loudness, changes to pain, fear and annoyance. I think I go up the slide. Yeah, that one. And that often the case, as you heard from Kenneth, that they're not necessarily separable from each other too. People can have issues with loudness as well as pain, among other things. That's kind of what I'm trying to get at with these gradients here. So it's a complex and heterogeneous phenotype, and it's also its underlying causes are probably more complex and diverse than we know too. So obviously, hearing loss is a major component, and many people that suffer a noise insult can develop hyperacusis. But as Wei alluded to, there's many neurodevelopmental disorders, Williams syndrome, and autism, as well as chronic brain disorders, fibromyalgia, and issues where a lot of these neurological neuropsychiatric disorders that have loudness tolerance or sound tolerance issues, right? And I think it's a really open question about how all these kind of different causes of hyperacusis are or are not related. When we talk about this, we think about this, should be thinking about this as a group of mechanistically distinct disorders that happen to have similar clinical presentation, or is there a convergence? You can go to the next slide now. On shared mechanisms at some level of function, right? And so what we're trying to do in our lab now is kind of embrace this diversity and this heterogeneity in hyperacusis, try to use it as a tool, as a strength, rather than as a weakness, and try to directly compare these different forms, different ways of inducing hyperacusis, different ways of measuring hyperacusis like behavior, and determine if there is convergence on shared mechanisms at some level of neuronal function. And we use animal models in our work. We use mostly rats, and I think the benefit of animal models is you have access to all these levels of analysis with, in-depth tools and very detailed assessment. But the challenge is you have to model a complex heterogeneous disorder in a rodent species, and that's not trivial. And so, some of the things we are trying to work on are, hit the button again, are developing models that have both this, you can go back, with both, construct validity, how are we inducing hyperacusis in the same way that it's caused in people? And so obviously knowing more about how hyperacusis arises in people is important for that, as well as face validity, that we have, behavioral readouts that on one hand are, adapted for a rodent model, but on the other hand, that hopefully can be translated into the clinical setting in people with human condition. Right. And so I'll tell you a little bit about some of the ways we've tried to gone about this. We developed several behavioral tasks that we think kind of get at different aspects of sound tolerance issues. We use an operant sound detection task. So this is simply training food restricted rats to poke their nose in a hole for a food reward whenever they hear a sound. It's a very standard kind of psychological tool that's used. And it's you can do, but the nice thing about it is it's pretty flexible. You can get a lot of information out of it. So we can use that to do some basic audiometric testing to look at, behavioral detection thresholds, hearing thresholds in these animals, which is very similar to the way hearing thresholds are measured in the audiology clinic. But what we also shown is that we can use the reaction time of the animal varies as a function of sound intensity. And we've done some solid controls to show that we really think this change in reaction time with intensity is at least a surrogate or a correlate of loudness growth in rodents. So that kind of gets at these more psychoacoustic aspects, the loudness, hyperacusis. But we want to try to get at some of the other aspects as well. So we have a different task, a sound avoidance task, that was developed in Dick Salvy's lab by Senthil Manohar, and it's essentially a place preference task where the animal can be in this dark enclosed area or this bright open one, and rats are nocturnal burrowing creatures, so they much prefer to stay in this dark enclosed area under normal conditions. But then as we introduce different intensity sounds into that dark box, we can see that their preference decreases as the sounds get higher and higher. So we think this is what we call this avoidance behavior, this decrease in dark preference. And we think this kind of gets more at the sound tolerant issue, the kind of effective response, how unpleasant or annoying those sounds are. So, I think we're making progress on some of these fronts. Especially absent up here is a model for pain hyperacusis. So I'm really excited to see your work, Megan. I want to talk to you more about trying to get that implemented in our rats as well. So we can really kind of cover our bases and look at these distinct kind of perceptual attributes of hyperacusis in these rodents. Right. Next slide. And then at the same time, we're trying to think of different ways that we can induce hyperacusis in our rodents, right? So obviously, looking at noise-induced hearing loss is a tried and true method, and as others have already alluded to, there's many different ways to induce hearing loss in animals, different types of noise exposures that often will have different effects on the cochlea, and so it's important to try to even within this category, they kind of separate these subcategories. So we looked at both long duration exposures that cause permanent threshold shifts and hair cell damage, as well as more kind of synaptotoptic exposures where you don't get permanent threshold shifts, but synapse loss. And we see evidence for hyperacusis-like behavioral changes in both these cases, but we see some key differences as well. So in the case of permanent threshold shifts, we see that the sensitivity arises more to low frequency sounds below the region of hearing loss, while with a kind of hidden hearing loss exposure, we see these sensitivities arising within the region of hearing loss more specifically. So even within this noise-induced hearing loss models, there's a lot of variability. And I think something important from the clinical side is to understand more about the kind of audiometric profile of hyperacusis. And Kenneth was talking a little bit about that from his personal experience, the types of sounds that really make what the frequencies are, things like that. Are there particular frequencies that are more unpleasant than others? And I should also mention that in both cases, while we see effects at the group level, it's really very heterogeneous across the population. Only a subset of the animals are actually developing these changes, just like as Megan said too. And so we've been able to show that by doing electrophysiological recordings from these same animals that are behaviorally trained, that the animals with the biggest change in their loudness growth or reaction times, in our case, were really well correlated with the animals that had the biggest change in their sound evoked responses in their brain, particularly in the auditory cortex, and to correlate better with cortical responses than subcortical responses. So we think this is one of the ways we can try to start pinpointing what areas we should be looking at in these models. All right, the next slide. And the other way we're going about this is looking at genetic models of autism, similar to what Wei Sun talked about in the first talk. And so we know, the reason why this is a interesting approach is because we know a lot about the genetics underlying autism. There's been a lot of these GWAS studies, genomic studies, and there's been now, I think, over 1,000 mutations have been implicated in autism. In many cases, they're fairly low penetrant. They only kind of increase your risk factor by a little bit, but there are a subset of these rare single gene syndromic forms of autism. You can see they only make up about maybe 15% of the entire autism population. But the nice thing about these disorders is that they're caused by a single gene mutation, so we can replicate it in animals very precisely, and they're highly penetrant. So if you have Fragile X syndrome or tuberous sclerosis complex, these are the two most common inherited forms of autism. You have over 50% chance of being on the spectrum, as opposed to something like a 5% chance in the general population. And we have rat models of both of these forms of autism, a efferent knockout rat and a TSC2 eager rat, and we are comparing them as well, doing some of the same behavioral tests, and we see pretty good evidence for both changes to loudness and avoidance behavior in the Fragile X animals. And we've now see that similar changes to loudness in the TSC animals, and we haven't done any of the other behavioral tests yet. Right. Next slide. Nope. Go back. You're going backwards. Yeah. Nope. There we go. All right. Yeah, it is. So, right. So and now, I think we're making good progress on these kind of two bookends. We can induce hyperacusis through a couple different ways, or hyperacusis like changes in a couple different ways, and we think we can model some of the different aspects of the perceptual components of hyperacusis. And now, what our goal is to kind of fill in these gaps along the way, using a variety of approaches. You can functional imaging to look at what are there differences in the brain networks that are activated by sound in these animals that are with evidence of hyperacusis. Using in vivo electrophysiology and cell type specific manipulations to try to get at the kind of microcircuit mechanisms that may be driving these changes in these areas, and then using ex vivo approaches to look at the kind of synaptic intrinsic properties that may be changes in those cell types that are altered, and eventually to the cell and molecular level to look at changes in molecular and gene expression that could be accounting for those functional changes that we observe. And so we haven't made our progress through this whole pipeline yet, but we're working our way down essentially. And so far, the thing that's probably surprised me most is we've seen more similarities than differences between our models, particularly both noise-induced hearing loss models and autism models. We see good evidence for cortical hyperexcitability. We see recruitment of non-classical auditory areas like the lateral amygdala. And we see changes to both the acoustic perception and kind of effective response to sounds in both these models. So maybe that's good news, and that maybe there are convergent mechanisms between these things, or maybe it means our way of teasing them apart are not good enough yet. So it could be a glass half full or half empty in that regard. But I think in any case, kind of profiling these different models at all these different levels of analysis, I think can be very informative about how different forms of hyperacusis are or are not the same. Thank you. Happy to take any questions.
Sorry, but excuse my ignorance on this, but I'm curious if patients have widely been fMRI scanned or anything else to look for similar findings to your rodent models there in terms of hyperactivity.
I think it's tricky to get hyperacusis patients in a scanner because it's loud, right? There are people, Fatima Hussein does a lot of good work. People people with tinnitus, but also with hyperacusis. So there are some studies out there, but they're not a ton of work. Yeah.
What about something like FNIRS or something that may not be quite as
Sorry, say again?
like FNIRS, like other types of functional scans that may not involve that kind of noise?
Yeah. No, I think that's probably a good idea to look at as well.
Nothing that you know of that's been done.
Not that I know of. No, I don't. We're just getting into the functional imaging. That's not really our bread and butter. So yeah.
Same.
Yeah.
Thanks.
No other question. Okay, thanks.
A lot of things that I click through.
I know.
Yeah.
No time to try to fix it right now.
Well, hello all. My name is Kat Weisz, and my lab is at the NIH and IDCD. So I really hesitated to take part in this panel today because I don't actually work on hyperacusis, but it got me thinking, well, why not? And so what I would like to talk about today is more some of the barriers to some of the experiments that some of us do and why doing hyperacusis research can be difficult. So this is really about animal models and having animal models hopefully be reliable, but for people who maybe don't do a lot of behavioral research and maybe have some age limitations on the kind of work that we can do. So we are really interested in neurons in the auditory system, mostly the lower auditory system, the efferents and the brain stem. So this is kind of a beautiful image of a slice through the brain, a coronal slice. Right now, we're working on the olivocochlear efferent neurons. So these are in a region of the brain called the olives. They send their axons to the cochlea, so olivocochlear neurons. These neurons will project out to the cochlea, and they can suppress the vibrations in the cochlea. So they're really important for hearing because they can shift the gain of our system between louder and softer sounds. They can protect our cochlea against noise-evoked trauma. They might be involved in attention. But it's also been shown that they could be hyperactive in tinnitus and hyperacusis. So we think that different neurons in the brain could be involved in these disorders, but we don't really know how or what changes at the single cell level. So can you go to the next slide, please? Yeah, so we are patch clampers. We do electrical recordings from single neurons. We're really interested in their basic electrical properties, their synaptic inputs. So how are they connected to the rest of the brain? And what are their inputs in the brainstem, which we think are much more diverse than has been previously appreciated. We're also interested in their outputs in the cochlea, which we also think are diverse. Now, this is fairly new research. It's only been recently that we've had electrical access to these neurons in mouse models. So we have to use genetic models that make the neurons fluoresce red to even find them and do these patch clamp recordings so that we can get a handle on what their electrical properties are like. So we're really in the state right now of trying to figure out just what are these neurons like normally, like in a normal developing mouse. What are they connected to? How do they respond to different electrical inputs? And so we're still at that point of figuring out their normal activity, but we would like to know how these neurons are acting, how they are changed in conditions like hyperacusis and tinnitus. But there are some barriers to this kind of research. So can you go to the next slide, please? So just a little bit about what we do. So this is what's considered kind of the standard reflex neuron pathway where you're getting sound into, this is my drawing ability. This is the cochlea. You're getting excitation into the cochlear nucleus and then to these MOC neurons, which are in the ventral brain stem. But we are now understanding with this research that it's much more complex inputs to these neurons. So just a little bit about what we've been doing for the past few years. You can probably click about 5 times. Yep. So we're showing they're getting inhibitory inputs from the MNTB. Other labs, so Larry Trussell's lab is also looking at these neurons. They're showing excitatory descending inputs from the inferior colliculus. And we know that a lot of their inputs are coming from the contralateral cochlear nucleus. And so some of the people in the lab that have done some of this work are Lester Torres-Canadius and Matt Fischel. Oh, and one more click, please. Yeah, and Kirpa Suthakar in the lab are also doing this work to look at the different parts of the brain that are impacting these neurons. And so it can be excitatory, inhibitory, modulatory. So there's a lot happening with these neurons and a lot that could potentially be changing in conditions like hyperacusis and tinnitus. And next slide, please. But we are doing these experiments. We're recording the electrical signals in a single neuron. And so these are hard experiments to do, but they're made especially difficult because they're in the part of the brain that has a lot of axons, a lot of white matter. And so just getting to these neurons to do these experiments is very difficult. So we're doing these in fairly young animals. We're getting maybe 3 weeks, 4 weeks old. And so this is a brain slice from a mouse. And you can see this is a 300 micron brain slice, and it's nearly transparent here. But thinking about how we could use hyperacusis models, it takes some time to evoke hyperacusis. And I think that it's still being debated. This is not my expertise, just what is the best way to evoke hyperacusis? Can we even do it in an animal that's young enough so that we can then take its brain and look at how these cells are acting? And so the age that we can do these experiments is a limiting factor right now. So we're looking at these neurons not just in the brainstem, but also in the cochlea. Can you please go to the next slide? And so right now we're focused on the outer hair cell region where these MOC axons are contacting outer hair cells, and it's their inhibition of the outer hair cells, which is how they're influencing the cochlear function. And there's also a circuit between these efferent axons projecting to the cochlea and the type 2 spiral ganglion dendrites, which other people like Megan has also worked on. And these are neurons that could be involved in pain responses. So we're interested in the MOC axons, how they're affecting the cochlea. And we've been looking recently at how they are directly interacting with the type 2 spiral ganglion neurons. Can you go to the next slide, please? And so this is another preparation where we are dissecting out the tissue from the mouse, although this happens to be a rat. And next, so I use a lot of animation, so I usually just kind of keep on clicking through my talks. We'll slice off part of the cochlea. This has been totally dissected from the bone. Slice off part of the cochlea. And then one more, please. Oh, and so these patch clamp recordings, I'm sorry. Oh, they're done in young rats and mice. And then here's an example of the experiments to record the electrical properties. We can do this in hair cells. So here are the stereocilia of some of the outer hair cells. And then we can remove some to access these type 2 neurons. This is the glass electrode that we use to perform these experiments where we're recording the electrical signals in the neurons. We can do this in the dendrites. We can do this in the hair cells. But this is 1 micron in diameter. And these experiments, we've really only been able to get up to like postnatal day 18 in a mouse. And a mouse will begin to hear at 11 or 12 days of age. And so going between 11 or 12 days and 18 days of age, that's not a lot of time to really evoke hyperacusis. And even if I knew how to evoke hyperacusis. So these are some of the challenges that we are facing in doing hyperacusis research in these animal models. Just the age makes it very difficult. So next slide, please. So these are questions that I would like answered so that we can move our work further. And I've already learned a lot of things in this room tonight. Thank you. I would like people in this room to tell me, how should I give a mouse hyperacusis? What is kind of the most direct way, the most reliable way? We don't really do a lot of behavioral testing. And some of these behavioral testing protocols, they take a while. They take animal training. We don't have time to do that because we need to operate in young mice. And I'm also curious, how do we give a mouse hyperacusis that is similar to the human experience? And are we able to really differentiate it from tinnitus? So these are all things that are not my expertise, but I would love to hear about from all of you. So really, I'm here because I would love to learn what the best way could be for us to advance our research about these neurons and other parts of the brain into hyperacusis research. So no data on which people can ask questions really, but.
I had a very nice presentation also reflecting on what you said also. I wonder, noise trauma can be a model, but noise affects a lot of different things too. again, I'm happy to explore the possibility of this glass knockout model to be something like that,
Mhm.
because it is really a condition that's quite unique. You have synaptopathy, you have normal hearing, you don't have tinnitus at baseline, but you have hyperreactivity to sounds. This is maybe an area where, you know, we can all work out on something.
Yeah.
Reflecting also on you know, your talk and yours as well. I always thought hearing loss was lagging versus other fields, you know, 20 years behind. I've been working at the Karolinska with other labs, trying to advance knowledge on pain, fibromyalgia, you know, things that are very also abstract. And we're not that far with hyperacusis as well. So, you know, the lagging time is really short behind. We have a lot of animal models and behavioral assays. You have your hypothesis, and Megan is developing those models. There is a fantastic network here, so you can establish animal guidelines, how to assess that in the animals, and get a crisp about what's happening. So
Yeah, that would be wonderful. Thank you. Talking about these genetic models also could be much more accessible and younger for these mice. Thank you.
Thank you. Okay. Can everybody hear me? Okay, good. I do have a loud voice. Just let me know if it gets too quiet by me dropping the mic too far. And while my slides are coming up, but they're up, that's great. Thank you. I want to say that I have first learned about hyperacusis about 15 years ago when I was at the Hearing Health Foundation, volunteering there with their emerging research grants program. And it was there that I met two individuals, one of whom was Brian Pollard, who started to educate me about the Hyperacusis Research International. And what they were really looking for was to find ways to fund research. And it was obviously a lot smaller then. And but we came to an arrangement where somehow they would transfer the funds to HHF, and HHF would act as a review organizing body to find the best grants that were out there to fund. And so it wouldn't have to set up a completely new independent logistic, you know, review body to review the grants. And this is really what I'm going to be talking about, is talking today about what I've learned from my experience with the Hearing Health Foundation and with my work since then in managing grants and managing research. So we're going to be focusing on hyperacusis research. So really the most important thing is to define the research mission. And I think that that this foundation has really done a good job in doing that. But there are several different definitions of hyperacusis and what kinds of hyperacusis there is. And so which ones do you want to focus on? But if you have a broadly stated mission, you'll also have attract a wide variety of proposals, and they're not often mission-centric. But if you have a very narrow stated mission, you'll attract fewer proposals and often of varying quality, but some could be really good and really fit the mission of the Scientific Advisory Board at the Hyperacusis Research Foundation. So the next slide is, and then the other side of it is the amount that the research awards are going to be. If they are limited award amounts, then you're more likely to attract emerging investigators, which again can lead to a wide variety of proposals, some which will be particularly focused and others which will be less focused, and some that are not even relevant, but they won't be so much. Well, really the biggest drawback is that these emerging investigators are going to be less experienced. So they're still earning their chops because they are still learning how to be investigators. They've finished their postdocs and they're learning how to manage a lab. If you have generous award amounts, you will attract many more investigators, but again, are also often less mission-centric. And so you need a much more rigorous review infrastructure to vet those applications to to make sure that they're going to do what you really want to fund. So the next slide, please. So, Hyperacusis Research is a newer, growing foundation, and it's building visibility, awareness, and impact. And so maybe one of the things that I was thinking about is to really maybe have an exhibit booth at ARO where people can drop by and begin to learn more about it. People who are just walking by, people who don't even know what hyperacusis is. And that way you can start to increase visibility and impact. It's going to be a multi-year thing. It's not something that's going to happen even in 5 years. You just have to put yourselves out there and be there. So next slide, please. So how do you attract high quality proposals? But this is particularly difficult. And so again, emerging investigators, they are great. They don't require large amounts of money. This is something that HHF has been doing for over 50 years, 60 years even. And so they now have alumni of their program who are now retired. And they have alumni who are at every single stage of their career stage. So they're not only young investigators, but they've gone on and got NIH funding, and then they've gone on and got really big dollar award amounts. But it takes 10, 20, 30 years to create that cohort of people who have that knowledge and experience and ability to write those big proposals and attract that funding. So if you're going to focus on the emerging investigators, those proposals will not only be less focused, but it takes time to build that cohort into experienced PIs who have really narrowed down the problem. And it needs a stable funding stream. You have to keep at it and keep at it. And this is something that HHF has done very well. And a way that I would frame that is that for every dollar that HHF has put in to funding this research, and this is Timothy, this is up to about 2014 before I left. I think it was calculated that for every dollar that HHF spent on research, that was an investment that recouped about $48 in subsequent research. And I'm sure it's more than that now. So it just keeps going up. So, yeah. So that's right. So if they put in $1 million, it brings in another $50 million into research. And that's how we have to think about it. So next slide, please. Established investigators, you might get to that result more quickly to what you want, but they need large dollar amounts to even think about writing a proposal for you. That's the challenge. And they need to understand the issue and what is going to be likely to lead to an outcome that is what the foundation, the research foundation here, and Hyperacusis Research really wants to do. So that's another big challenge. And they're going to want that kind of money for more than 1 year, like in ARO1, we're talking about $250,000 a year plus indirect costs. And that's on top of the direct costs. And for HHF, they don't allow much in the way of indirect costs to go. So these, because PIs who want multi-year funding, their institutions want those indirect costs. And so you're talking about another 50% on top of whatever that award amount is. So next slide, please. So again, Hyperacusis is a newer growing foundation. So we have to manage expectations of how long you can keep going forward and how long you can fund this process. And so the mission goals should be tempered by the kind of funds that are available. And this is something that I thought that Christopher really brought up earlier, is to consider a consortium model of both newer and established PIs working together, working with a limited amount of funds. But by working together, you can create a much more effective, impactful direction of research together, instead of all working independently in silos. And that, again, is something else that HHF is doing with their hearing restoration project. And I've witnessed the impact of that And it really has led to a lot more advances between that consortium of PIs. So next slide, please. So the logistics to fund a meritorious proposals. HHF was really instrumental in teaching me this. And now that I lead a program where we fund a center and we have our own funding programs for investigators within the center, I've really come to appreciate how we need a program officer to disseminate calls for proposals, to answer queries, to solicit applications, and to really get that process going. It's very time consuming. And then you also need independently a scientific review officer to recruit the reviewers. This is somebody who knows the science and will know the reviewers who can review those proposals. And they have to respond to applicants, assess conflicts of interest. Could be a part-time position within a larger organization. And HHF did have those people when I was there. I'm sure they still do. And I know Dan Tollin, and he really is the scientific review officer, and he picks the chair, and that person runs the meeting. And I've had to do the same thing within the Center for our own internal proposals. So the experience I got from HHF has really helped me. So I really want to thank your organization, Timothy, for all that support. So I think if you have to find a dedicated program officer and SRO, and it costs money, but more importantly, it costs time and effort and dedication. And that's something that I think is a really worthwhile effort, because that way you get the applications that you really want to fund to do that research. So on the next slide, please. So I really thought that all those years ago, 10, 15 years ago, Hyperacusis Research partnering with HHF was a smart initial step to fund those research projects. And I really think that the next steps is to focus on building dedicated logistics to deliver scientific advisory board preferred mission-centric goals. And then you tailor those to the amount of funds that you are able to disseminate for that particular call for applications. So it sounds simple. It is simple, but it takes a lot of effort just to manage it all. It's a lot of people management is involved. So so next slide, I think that's the last one. So yes, we do have this one. Yes, so Hyperacusis has a dedicated fundraising team to increase that capacity for the research that you want. So recruit those prospective donors that align with your mission-centric targets. Potentially recruit other foundations and centers that support mutual goals. So you can talk with them for what you really want to do and see if there are ways you can even do contracts. That's what NIH does when they want a really specific goal focus for. Instead of a research grant that is reviewed by a review committee, and really increase visibility. And it sounds easy, but it isn't easy. It takes time and focus and people who are willing to do it. No one person can do it all. So in a way, you have to spend money to raise money in order to fund, in this case, Hyperacusis Research. I think there's one more. So there's risks and rewards in all of these activities. And one thing that I really did learn is that, and not particularly at HHF, but one singular big donation doesn't help. You need you need a diverse funding stream, just like we all do for our retirement plans, so that if one thing comes down, another thing is doing better. And so you get this stable funding stream. A big singular donation is good, but it can lead to problems. And I remember this particularly after the housing crash in the United States in 2008. And there was a huge funding stream for NIH, which was great for 2 years. and it funded a lot of new research. But that research all had a limited time span of about 2 to 5 years. And then that money went away. And there was a lot of pain 3 or 4 years later, and a lot of researchers couldn't get the money and the funding to keep those research projects going. And so it caused a lot of pain. So you really have to watch out from having a big singular donation and using it on capital projects. You have to invest it to make sure that there's a steady funding stream so that people know that the money is always going to be there, not that it's going in cycles, because scientists and science takes time. So therefore, they're kind of risk averse to changes in those funding streams. So, really, that's really what I wanted to share about the challenges of funding a research organization rather than the research here. And I think you have a great group of people here who can all work together to create a consortium or at least a discussion group to decide what kind of research is going to get to the goals of Hyperacusis Research International. I think that's the last one. Can we just check? Okay, this is yours. Thank you.